Hello, my name is Adia and this is the next episode from Curion on Genomics. This will also be visible in the biological um, sciences event at Youth STEAM Advisors. So first I will describe what genomics is and describe the difference between genetics. I will also describe the difference between functional and structural genomics. And I will also explain about how a genome defines who we are, and also like what codons and introns and exons are. Then I'll also talk about gene mapping and like how it is different from gene sequencing. And I'll also talk about like gene editing a little bit and how genomics can and the connection between genomics and diseases. And then I will lastly discuss like how genomics can be influenced by AI in the future. So now we can start with the first topic and understand what genomics is and how it is different from genetics. So it's important to understand where the word came from. So the word genomics originates in 1986, created by geneticist Thomas H. Roderick. He proposed it as a name for a new scientific field that is focused on the study and comparison of genomes across species. So this term combines genome with the ISC suffix, like techniques, which refers to an application, skills, or techniques. The genome itself, from which genomics is derived, was coined by Hans Winkler in 1920, which is merging Greek words genesis, which means birth, and soma, which means body. So basically it means the body of genes. So in simple terms, a genome is the operating manual containing all of the instructions that help us to develop from a single cell into a human being. So it includes the complex set of genetic information. This information is held by deoxyribonucleic acid, which is packed into structures called chromosomes. This is a chromosome, and this is deoxyribonucleic acid, which is also DNA. And each chromosome contains a stretch of genetic code, which are called genes. So the genes are actually only paired A, T, and C, G, like apple to tree and car to garage. So humans have 25,000 genes, and each one gives an instruction to like how the body to the body. So humans usually have like 2 copies of each gene. One copy is inherited from each biological parent, and there are usually 23 pairs of chromosomes in each cell of the body. So this is all in one cell, and then this DNA is in one chromosome. So the entire amount of DNA in the cell, including all 25,000 genes and the non-coding part of the DNA, is totaling around to 6 billion genes plus the non-gene information. That is called a genome. So another analogy to understand it would be that if the genome is an instruction manual, a chromosome would be a chapter. So genes would be sentences in the chapter, and DNA represents the letters that make up each word. Now let's try to understand the difference between genetics and genomics. Genetics is the study of genes and their effects one at a time. Genomics is genetics in a larger scale, the study of many or all of the genes. DNA is the molecule of life containing the information that defines and makes us. In eukaryotic cells, like human cells, most of the DNA molecules are stored in the nucleus, organized into chromosomes. Some DNA, um, material is also stored in the mitochondria of the cell. So I will discuss the structure of the DNA molecule and how it is organized in depth in the next topic. So now let's just remember that genomics is the entire set of DNA information. This information consists of both the coding and the non-coding parts of the DNA. The coding part of the DNA is what is termed genes and is the study of genetics. So genetics is the study of heredity in an individual. Structural genomics focuses on the physical structure and organization of the genomes, including sequencing and mapping, while functional genomics investigates gene expression and function within a genome. So essentially, the structure of genetics provides the blueprint, while the functional genomics explores how the blueprint is used. So one way to understand the difference between structural and functional genomics is to consider the structure of structural genomics as how books are organized on a bookshelf, and functional genomics is how one book on a bookshelf will characterize or impact you. So now let's try to understand why the study of genomics is very important. So to understand this, we also have to understand how the genome is structured and what the coding and non-coding parts of the DNA are. So we will first see the structure of DNA molecule. Remember, the DNA molecule is a double helix shape, and it contains the genomes and is stored in a highly coiled in the 23 chromosome pairs in humans. A DNA molecule is composed of two long strands. A DNA molecule is composed of two long strands, each of which is made of building blocks called nucleotides bonded together. Every nucleotide is made up of three components: a phosphate group, a deoxyribose sugar, and a nitrogen-containing bases, known as adenine, thymine, cytosine, and guanine. Bases are often referred to by the letters A, T, C, and G. So A only goes with T because an apple goes on a tree, and C only goes with G because a car goes in a garage. The double-stranded molecule is twisted into a double helix shape, resembling a twisted ladder, where the rails of the ladder are made up of the phosphate and the deoxyribose sugar, and the rungs are made up of nitrogen-based pairs. Opposite strands run anti-parallel to each other, meaning that they run in opposite directions. This ensures that the strands fit tightly together. Our genome can be divided into two parts: the parts that code for proteins and thereby for our traits, called the genes, and the parts that do not code for proteins, which are called non-coding DNA. The intergenic regions, which makes up most of the human genome, includes introns, regulatory sequences, telomeres, and satellite DNA, among others. While they don't directly code for proteins, non-coding DNA plays crucial roles in gene regulation, chromosome structure, and other essential cellular processes. It is not junk DNA as it is long believed to be. Therefore, a DNA strand is made up of genes and the intergenic region. Within the gene, there are two regions: the exon, the region of the gene containing the coding sequence of nucleotides. The other region inside the gene is called the intron, which is a non-coding sequence within genes. The non-coding intergenic region contains regulatory sequences, which include promoters and enhancers. These control gene expression by influencing where and when a gene is transcribed. In addition, the telomeres in the chromosomes, which are protective caps at the ends of chromosomes that prevent DNA damage and chromosome degradation, also make up the non-coding part of the genome. Some of the non-coding repetitive DNA sequences are found in some other regions of the chromosomes as well, particularly near centromeres, and are called the satellite DNA. Now, how does the genetic code turn into a protein that defines our traits? The flow of genetic information from a gene to a protein follows the sequence DNA to RNA to protein, a process often referred to as the central dogma of molecular biology. This involves two main steps: transcription and translation. In the process of transcription, the information encoded into a gene is copied into a messenger RNA or mRNA molecule. This process occurs in the nucleus of the cell. This is the first step where the cell copies a gene's DNA instructions into a similar molecule called RNA. So it's basically like copying a recipe from cookbook. An enzyme called RNA polymerase reads the DNA and builds the RNA strand, which happens in the nucleus. So one important thing to remember is the process called splicing. So the splicing process involves the removal of introns and the joining of exons to create a mature mRNA, which is ready to travel out of the nucleus to ribosome. Next, in the process of translation, the mRNA molecule then moves to the ribosome where the genetic code is read and the corresponding amino acids are assembled into a protein. In the second step, the mRNA recipe is used to build a protein. The ribosome reads the mRNA codons by one matching each to an amino acid brought by transfer RNA or tRNA. These amino acids link together to form a protein, like assembling ingredients into a dish. Essentially, the DNA acid acts as a template. The mRNA carries the instructions, and the ribosome translates those instructions into functional protein. So you might have heard of a codon. So a codon is a set of 3 nucleotides in DNA or RNA that acts like a code word. So each codon tells the cell which amino acid to add a protein, to which amino acid to add to a protein, or signals to start or stop building it. So there are a total of 64 codons. So now let's investigate gene mapping and gene sequencing, what they mean, how they are different, and why they are important. So what exactly is gene mapping? It's the process of determining the location and order of genes on a chromosome. It's like creating a map that shows where specific genes are positioned along the DNA strands. As shown in the picture on the right, gene mapping helps scientists to understand how the genes are inherited and how they interact. How does heredity work? Role does crossing over play during meiosis, and so on. Gene mapping also helps to identify genes linked to traits or diseases like familial ALS, and then to develop targeted treatments or genetic tests. Another aspect you should try to understand is the nomenclature used in gene mapping. As you can see in the picture on the left, there are 2 main arms of chromosome. Note that a chromosome is made up of 2 homologous chromatids pairs. The longer arm is called the Q arm and the shorter arm is called the P arm. So now that the chromatid is divided into regions, the numbering starts from the centromere and goes into the telomere. Within a region, there are further divisions called bands and then subbands. Therefore, 3p22.1 would identify the location of a gene located on the 3rd chromosome, on the smaller P arm, in region 2, in band 2, and in subband 1. So let's now look at gene sequencing. Gene sequencing is the process of determining the exact order of nucleotides, A, T, C, and G, in a segment of DNA or an entire genome. It's like reading the complete instruction manual of a gene or organism to understand its genetic code. Gene sequencing can help identify mutations or variations linked to diseases, study evolutionary steps and enable personalized medicine by analyzing an individual's genetic makeup. In short, gene sequencing decodes the DNA sequence, providing a detailed map of genetic information. Therefore, the main difference between gene mapping and gene sequencing is that gene mapping identifies the locus or location of genes, um, and uh, um, and their relative distance with the genome, whereas gene sequencing spells out the order of the nucleotides, which makes up the genes in the genome. Furthermore, gene mapping results in a less detailed outcome, while gene sequencing results in a fully detailed outcome. Gene editing is a technique that allows scientists to precisely modify an organism's DNA by adding, removing, or altering specific genes. It's like using molecular scissors and a paste tool to edit the genetic code. The most common method is CRISPR-Cas9, and it uses a guide RNA to target a specific DNA sequence, and an enzyme, Cas9, cuts it, enabling changes to be made. This technology can help correct genetic disorders like sickle cell disease, which is when some of the cells in the body, some of the red blood cells become shaped like crescents, and it blocks the blood flow, so oxygen cannot get to all the bodies in the cells. Enhance crop resistance or livestock traits, and study gene functions in research. It's a powerful way to rewrite DNA for health, agriculture, and science. Now, I will talk about CRISPR. So the development of CRISPR genome editing has revolutionized scientists' ability to edit the genome. In 2012, research by IGI founder Jennifer Doudna, Emmanuel Charpenter, and their teams developed a method of repurposing a bacterial immune system called CRISPR, an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats, and to make breaks in DNA at precise locations using CRISPR-associated protein or Cas9. Cas9 is like molecular scissors to cut DNA. When DNA is cut, this creates an opportunity to change the DNA sequence at and around the cut site by harnessing the cell's natural repair pathways. Now I will close by discussing the future of genomics and how AI could help in the field. The image from the WHO website visually summarizes the impact of genomics on healthcare. It highlights some of the key themes that would predict the future of this field. Genomics will transform from a research field to a medical tool, especially with recent advances like CRISPR and Cas9. It will pave the way for personalized medicine, tailored treatments, disease prevention, early detection, and even pathogen surveillance and tracking outbreaks. Now imagine the possibility that AI could help as a smart assistant to researchers and scientists in this area. With its advanced computing power, AI can analyze massive amounts of genomics data, find patterns or changes linked to diseases like cancer or rare conditions. AI tools can quickly analyze genomic sequences to spot genetic issues, predict diseases, and speed up diagnosis and personalized treatments. So in conclusion, the future of genomics promises revolutionary change in many areas of life. Advances um, in sequencing will make genetic testing faster and more affordable, bringing personalized healthcare with everyone's reach. Treatments will increasingly be tailored to individual DNA, while gene editing tools like CRISPR may offer cures for previously untreatable genetic diseases. AI and big data will help uncover hidden patterns in our genomes, leading to new insights. Beyond human health, genomics will improve crops, aid conservation and drive innovation in medicine and biotechnology. However, with these breakthroughs come important ethical challenges about privacy, fairness, and responsible use. I hope you enjoy. If you like this topic, then you can join events at YSA to learn more about other topics as well, and the link will be in the video. Thank you.